Welcome back, and we'll be continuing with the classification of kingdom Protista. We have already looked into the fungal protists and algal protists. Almost like 8 phyla we have discussed in the kingdom Protista. The next 10 that comes under animal-like protists, or what we generally speak as Protozoa. Among the Protozoa, we'll be looking in detail the characters of all those phyla given in red, that is Phylum Rhizophora, Coeliophora, Parabasilia, and Sporozova with an example. Okay, so the rest of the phyla, since we are not supposed to look into in detail as per the syllabus, we'll just see what are the major characters and what is the ecological importance and a few examples. Okay, so now animal-like protists, or what is referred as the Protozoans, they, as the name suggests, they are animal-like, so they don't have chlorophyll. Unlike the algal protists, they are chlorophyll lacking. Hence they are non-photosynthetic. And we can see free living forms, parasitic forms, symbiotic forms. All these can be found in these protozoans. Among this one, free living forms, most of we can see them in freshwater, in marine ecosystems, and moist soil, etc. Now, the first one we will be discussing is Phylum Rhizophora. It is otherwise referred as Sarcodina, and the very familiar protist which you have been studying from your very lower classes, that is amoeba. It comes under Rhizophora, and we'll be looking in detail the characters of Rhizophora and the example. Now, foraminiferans or Phylum Foraminifera, they are otherwise referred as foraminiferans. They include a group of amoeboid and mostly marine protists, and they have branching and anastomosing pseudopodia. Okay, so their locomotion, since it is amoeboid, they are pseudopodia. It is a locomotory structure, but it is branching kind of a pseudopodia, which is referred as Reticulopodium. Okay, and they have a network of branching cytoplasmic strands and chambered shell. Okay, that is, body is covered with a shell which is chambered and it is porous. Okay, and in majority of the foraminiferans, we can see this shell. It is made up of calcium carbonate and silica. That is, the shell. Body is provided with a shell, and this shell is chambered and it is porous. That is, it bears pores on that. And this shell, in most of the foraminiferans, we can see it is calcareous in nature. That is, it is made up of calcium carbonate and silica. So what happens is, once these foraminiferans die off, these calcium carbonate or the calcareous shell, it forms a fossil deposit. Just like what we saw in the case of diatom, here also we can see that this calcareous shell, it drops down and it forms a foraminiferan ooze. Just like diatomaceous earth, the foraminiferan shell, it forms a fossil deposit. Okay, so that is one of the most important feature of foraminifera. Another feature which we have already seen, it is a pseudopodia, amoeboid structure, amoeboid organisms, and hence they have pseudopodia. But the pseudopodia, as we have already mentioned, they are branching and anastomosing. Anastomosing means branching, okay, branching and rebranching, etc. So it forms a very extensive branch structure, and that kind of a pseudopodia, it is known as a reticulopodia. Okay, and this reticulopodia, it is used for locomotion, for anchoring onto certain substratum, anchorage, food collection. Okay, and these pseudopodia extend out through the pores on the shell. We have already seen the shell is porous, isn't it? These reticulopodia, it extends out through the pores on the shell. One example which we can see is the Elphidium. Okay, this Elphidium, it looks like a molluscan shell, but it is a foraminiferan. It is a protist. Okay, a very smaller microscopic organism. Now, very unique feature. Another unique feature regarding foraminifera, it is about their kleptoplastidy. Okay, klepto means stealing, right? It is stealing in Greek. Okay, so kleptoplastidy, it is actually a symbiotic phenomenon wherein before we go into that, we can see that the foraminiferans, they feed on, many of the foraminiferans, they feed on algae. Okay, so when they feed on algae, they feed, they ingest the algae, keeping their chloroplast intact. Foraminiferans, they feed on green algae, for example, they feed on zooxanthellae, zoochloralae, etc. And when they feed on them, they keep their keeping their plastids or chloroplast intact. They feed everything else. They break down everything else. Okay, so what happens is these plastids from the algae, they get sequestered or they get collected inside the host organism, and this actually give like a this kind of a symbiotic phenomenon is what is referred as a kleptoplasty, and this is shown by many of the foraminiferans. Okay, as we have already seen one example, the Elpharium, right? Now the next group, it is Actinopoda. Okay, Actinopoda, and again this is also, so you can see this is Rhizophora. It includes amoeba, so it contains pseudopodia as a locomotory structure. Here also it is amoeboid protist, and this is also include a group of amoeboid protists. Okay, that is Actinopoda also. Unlike foraminifera and Rhizopoda, Actinopoda do have a specific kind of pseudopodia which is referred as axopodia. Okay, you can see over here, very sharp, very pointed, isn't it, slender. So these structures are known as what you call axopodia. And this Actinopoda, it includes two groups, that is Radiolaria, which are completely marine, planktonic actinopods, and heliozoans, which includes freshwater planktons. Okay, so marine planktons are included under Radiolaria, and freshwater planktons are included under Heliozoa. So Radiolaria and Heliozoa are two groups of Actinopoda under phylum Actinopoda. All the members of phylum Actinopoda have a spherical body, as you can see in this case. Sorry, yeah, in this figure, almost like spherical body. And you can see these, they have very long, stiff needle-like. These are the pseudopodia. Okay, long, stiff needle-like pseudopodia, and these are what is referred as axopodia. And these axopodia, they emerge through the openings on the outer covering, the test. Okay, so in most of the Actinopoda, we can see that the cortex and the medulla, that is the internal part and the outer part, it is separated by an organic capsule, and this capsule is what is referred as a test. Okay, so these axopodia, it emerge out through the test, the pores on the test, the openings on the test. Okay, and these axopodia, they are used for locomotion, for anchorage, just like in the case of what you have seen in the case of Elpharium or foraminiferans, Reticulopodia. Here also it is used for locomotion, anchorage, food capturing, and all those purposes. Okay, and in the what you call flotation on the water surface. What happens is they contain a large number of tiny non-contractile vacuoles inside the cortex, and which helps in increasing buoyancy. So apart from this, these what you call axopodia also help in keeping it afloat. Okay, so this is about the phylum Actinopoda. One example is Actinophras, right? So Celiophora, as the name suggests, it has a cilia. It has cilia as a locomotory structure. So all these three, Rhizopoda, Foraminifera, and Actinopoda, had pseudopodia as their locomotory structures, while Celiophora, it contains cilia as their structure, locomotory structure. And obviously, very best example is Paramecium, which we have learned in very detail. And another example, you will be learning it in the coming presentations. The next group is phylum Parabasalia. Again, it includes protists which are flagellate. Unlike Celiophora, it is a flagellate, and they are anaerobic. And we will be dealing it in detail in the coming presentations. Okay. The next we have is phylum Opalinata. Phylum Opalinata, it is cilia-bearing protists. They are large size comparatively, and they are endosymbionts. As the name suggests, endosymbionts means it lives inside the other symbiont. Okay, and it usually inhabits the colon, rectum, or cloaca of fishes, amphibians, and reptiles. Okay, Opalinata, the example is Opalina. We have here. Okay, one example. It is an endosymbiont in frogs. Okay, so it is endosymbiont, so it doesn't cause any pathogenic effect on the host body. Now, body is large size and usually flattened with a ciliary covering. So all these, you can see the cilia over here, isn't it? Okay, so these are ciliary bands, and you can see it is surrounded with cilia. Then intraciliary apparatus and alveoli are absent. We have already studied that in Ciliophora, in Paramecium, we have mentioned intraciliary apparatus. We had seen that is the pellicle, and alveolar layer we had. Such kind of things are absent in the case of Opalinata. And there is a pellicle is covers the body, all around the body, and it's supported by microtubules. Unlike Ciliophora, you can see ingestatory apparatus is also absent in the case of Opalinata, and the nuclear apparatus is monomorphic. That is, it consists of two or more nuclei, but all these nuclei are identical. That is why it is referred as monomorphic, unlike dimorphic in the case of what we call the Paramecium, which we have studied, macronucleus and micronucleus. Such kind of a differentiation is not possible in the case of monomorphic nuclear apparatus, even though they may have more than one nuclei, but it may not be able, we may not be able to differentiate based upon the size. That is why it is known as a monomorphic. Okay, example is Opalina, which is an endosymbiont in frogs. The next phylum, it is Phylum Sporozova. And the Phylum Sporozova includes parasitic forms, endoparasitic forms, and you will be learning it in detail with example later. The next one we have is Phylum Metamonada, and it includes anaerobic flagellate Protists. Okay, it includes flagellate Protists. They don't have mitochondria and plastids. Most of them are endosymbionts, and we can see even parasitic forms. Okay, very uniquely, you can see one example here. Okay, Giardia, right? They have oval and elongated body. You can see oval body, right? And it is covered by pellicle, and you can see here they have a flagella, right? Here you can see these are flagella, okay, which helps in their locomotion. Now, these doesn't have mitochondria or plastids, but they do have hydrogenosomes. Hydrogenosomes, it is considered to be relics of mitochondria. That is, mitochondria once present, it may have reduced to form what is known as the hydrogenosomes. Okay, and these structures just similar to an eye, isn't it? Okay, these are the actually nucleus, right? So it may contain one or more pairs of nuclei, and hence it is Giardia. It is otherwise known as grand old man of the intestine, because it's when you see the structure, it looks like an old man, the face of an old man. Okay, so it is a parasite, and it resides in the small intestine of man, this Giardia. Okay, and coming back to Metamonada, we can see that it is a phagotrophic nutrition or parasitic. Some forms also produce cysts. Okay, since they are what you call anaerobic and endosymbiotic, also cyst formation is also seen in some of the this one, what you call some examples. Okay, so this is about the Metamonada. The next one is Phylum Kinetoplaster. Okay, it includes a small colorless naked parasitic flagellates, and they have kinetoplast and 1 to 3 flagella. The kinetoplast, the what you call Kinetoplasta, it is they have a elongated body. You can see here with example Trypanosoma. Okay, very elongated body, narrow with or without pellicle. It may contain 1 to 3 flagella for their what you call locomotion, and they are like a undulating membrane. It is a unique feature in some of the kinetoplasts. Then centrioles and plastids are absent. Then another important feature is that with respect to the trypanosome, okay, trypanosoma, and there are plenty of other examples. Then Leishmania, they are parasites in humans. Okay, Trypanosoma gambiense, I suppose you are familiar with that. Okay, the Trypanosoma gambiense, which is parasitic in humans. Okay, now the last group, unfortunately, we have to pass on to the next one, the Phylum Kinetoplaster. They are sessile, solitary or colonial. This is a colonial form here. Okay, flagellates with a vase-like body. You can see funnel-shaped or vase-like body, and they have a like a flagella. they are have encircled the base of the flagella. So this is with regard to the choanoflagella.